Hey everyone, welcome to AP Help 101. So this week I'm going to be talking about the cell and really parts of the cell and what happens inside the cell. Prokaryote versus eukaryote. So in a prokaryote, there's no nuclei and no internal membranes. So there's really no actual nucleus in a prokaryote, and that's really the main difference between a prokaryote and a eukaryote. They also have a nucleoid region, however, which is where the chromosomes are located. And the chromosome is actually just a really large circle. It's not coiled up into chromosomes that we see in human cells. It's just one large circle. And a good example of a prokaryote is bacteria. Now, a eukaryote has a nucleus and membrane-bound organelles. Its nucleus is bound by a double membrane, which would be the nuclear envelope, actually, that's the correct term. And DNA is wound up into chromosomes, just like the ones that you see, like a human chromosome, they're wound up into chromosomes. And for an example, it'd be immune cells, which is found in our body. Of course, our body's made of eukaryotic cells. So this is what a prokaryote looks like, and this is what a eukaryote looks like. So a prokaryote has very few things in it, actually, inside of it. It's just some cytoplasm, a large chromosome, or other things, but the eukaryote is very complex. So, the prokaryote probably existed before the eukaryote did. So we're going to talk about organelles of the cell, and this is kind of the agenda for what we're going to talk about. So we're going to hit the nucleolus, ribosomes, peroxisomes, nucleus, the ER, endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, vacuoles, chloroplast, cytoskeleton, centrioles, centrosomes, and microtubules, a cell wall, and a plasma membrane. So there's a lot of organelles in the cell, and most of them pertain to both an animal cell and a plant cell. But sometimes certain organelles are only found in one or the other, so I will, of course, say if it's in both or not. So we're going to talk about the nucleolus first. So this is where ribosomal RNA is synthesized, and it's a tangle of chromatin and unfinished ribosomal precursors, basically. And the nucleolus is found inside the nucleus. So actually, the nucleolus takes that chromatin and unfinished ribosomal precursors and synthesizes it into rRNA or ribosomal RNA. So we're going to talk about ribosomes. They're basically your protein factories of the cell. They're found either floating in cytoplasm or on the rough endoplasmic reticulum. So they play actually two different roles depending on where they're found. If they're found on the ER, they're usually used for exporting things out of the cell. And if they're found free-floating, then they're more likely used for protein synthesis. So peroxisomes, they contain catalase, and that converts hydrogen peroxide into water by just taking out some oxygen. They also detoxify alcohol, and peroxisomes are actually found in both plant and animal cells. The nucleus. The nucleus contains chromosomes, so this is where the DNA is located, actually. It's covered by a nuclear envelope. This nuclear envelope has pores, which helps in the transport of molecules in and out of the nucleus. So this nuclear envelope is just a double membrane, basically covering the nucleus, and it really it controls what goes in and what goes out. So we don't want the wrong things going into the nucleus or the wrong things coming out. The nucleus contains the instructions for how each cell functions. So that's actually the DNA. And that's why I like to call this the ruling government of the cell, because the nucleus basically tells all the other organelles what they're supposed to do and how to do it, when to do it, things like that. Endoplasmic reticulum. There are two types of endoplasmic reticulum. We have rough ER, which has ribosomes and makes proteins. So you can tell the ribosomes are used in protein synthesis, so you can remember that the rough ER makes proteins because it has ribosomes. There's also smooth ER, which synthesizes steroid hormones. It stores calcium ions to help muscle contractions, and it detoxifies drugs and poisons. So the smooth ER has a lot of different functions comparatively to the rough ER. The Golgi apparatus. This processes and packages some substances made in the rough ER. It also secretes substances to other parts of the cell. I'd like to call this the cell's postal system because it really is just a transporter of materials everywhere, all throughout the cell. Lysosomes. Lysosomes are sacs of digestive enzymes, and this is where intracellular digestion takes place. So lysosomes help the cell renew itself by recycling cell parts. And they're usually not found in plant cells, they're more abundant in animal cells. I like to call lysosomes the waste treatment for the cell because it just gets rid of things that the cell doesn't need, but it recycles parts, so it treats the cell and gets rid of waste. Mitochondria. This is where cellular respiration takes place. This is where you get all of your ATP and all of your energy, basically. Mitochondria has an outer double membrane and an inner membrane with folds called cristae. Mitochondria actually have their own DNA, which you get from your mother. And so this is the reason why scientists think that mitochondria and chloroplasts were once free-living prokaryotes. I like to call mitochondria the powerhouse of the cell because basically this is where you get all of your energy. This is where cellular respiration takes place, and with cellular respiration comes energy. Vacuoles. Vacuoles are used for storage. Plant cells usually contain one large central vacuole, whereas animal cells usually contain many small vacuoles. Actually, some protists have contractile vacuoles which pump out excess water. So that's just an example of what a vacuole can do. That's just in protists, though. Vacuoles are usually used for storage in plant cells and animal cells. Chloroplast. These are found in plant cells only. They contain a green pigment called chlorophyll, which takes in light energy and makes sugar. This is a part of photosynthesis. This is where photosynthesis takes place. Chloroplasts have an outer membrane and an inner membrane called thylakoids. These are also considered to be their own prokaryote, just like the mitochondria. Cytoskeleton. A cytoskeleton has many roles. It can maintain cell shape, controls the position of organelles, is involved with the flow of cytoplasm, and anchors cells in place. There are multiple parts to it, actually. There are microtubules. These are made of tubulin, which makes up cilia and flagella, which are used for movement. The cilia are little strands on the surface of the membrane, and those also help with movement, as well as the flagella, which is like a tail at the end of the cell. They follow a 9+2 configuration, which is the configuration of the cilia and the flagella. And then microfilaments are assembled from actin filaments, and they help support the shape of the cell. Centrioles, centrosomes, and microtubules. They lie outside the nuclear membranes. They organize spindle fibers, which actually I believe you can find in mitosis and meiosis. Two centrioles together make up a centrosome. Plant cells do not have centrosomes, actually. Centrosomes are just found in animal cells. However, plant cells have MTOC, microtubule organizing centers. Cell walls. This is, again, only found in plant cells, not in animal cells. They're made of cellulose in plants. Fungi cell walls are made of chitin. They help with the structure of the plant, make sure it's straight, strong. Some plants actually contain a secondary cell wall. Not all, but some do. Secondary is right behind the actual cell wall. We're actually going to talk about the plasma membrane. This is the police force of the cell, I like to call it. It's selectively permeable, and there are many proteins found on this membrane. So there are glycoproteins, glycolipids, there's all sorts of things found on the membrane. It contains it's basically a phospholipid bilayer. It has a hydrophilic head and a hydrophobic tail. And it just really, it decides what goes in the cell and what comes out. And that's really important because we don't want to bring in anything bad into the cell, and we want to release anything that's bad from the cell. So the plasma membrane really plays an important part. And here are a couple pictures of an animal cell and a plant cell. So we pretty much went over most of the main organelles that are found in a cell. As you can see, there are many of the same organelles in an animal cell and a plant cell, just a few differences. Plant cells have chloroplasts, and they have a cell wall. And animal cells, they they don't have the cell wall, but they do have the centrosome, which the plant cell doesn't have. So there are a lot of differences. You can take a look at each picture just to see there are actually 4 different parts to a nucleus, but we did not go over all of them. But you can take a look and just see the differences between the animal cell and a plant cell just to get to really know them. Now we're going to talk about transport. So there's passive transport, which is the movement of molecules down a concentration gradient from a region of high concentration to a region of low concentration. So basically it's kind of going with the flow, sort of. Passive, just going with the flow, not going to go against anything. There are 2 types of passive transport, actually. The first type is diffusion. There's 2 types of diffusion on top of that. There is simple diffusion, which does not involve any protein channels. The substance just diffuses in without any help required. For example, oxygen, gases like that, they don't need any protein channels. Then there's facilitated diffusion, which requires a protein channel to move the substance, but not necessarily any added energy. Then there's osmosis, which is just a specific type of diffusion for water. Osmosis describes the movement of water, whereas diffusion describes the movement of particles. Then there's something called a countercurrent exchange, which is the flow of adjacent fluids in opposite directions that maximizes the rate of simple diffusion. And you can see this in gills for fish. So the second type of transport is active transport, and that's the movement of molecules against a gradient, and it requires energy, usually in the form of ATP. There are actually many different examples of active transport, but here's a few. There are usually pumps or carriers. So a couple examples are the sodium-potassium pump, which pumps ions across the nerve cell membrane, and the electron transport chain, which pumps protons across the membrane. So those are examples of active transport. There's also exocytosis, in which a nerve cell vesicles release neurotransmitters into a synapse. That's another example. There's penocytosis, which is cell drinking, where a membrane surrounds just a small particle and brings it into the cell. But then there's phagocytosis, which is cell eating, and that's engulfing large particles of the cell. And finally, we're going to talk about different types of solutions, and not math solutions, but solutions of solvents and solutes. So there's a hypertonic solution, which there's more solute on one side than the other. There's a hypotonic solution, where there's more solvent on one side than the other. And there's an isotonic solution, which is equilibrium. Same amount of solute, same amount of solvent. And a solute is actually the particle being dissolved into the solvent. So for example, a solute is sugar, whereas a solvent is water. And this is just a picture of what each of the different solutions looks like. So you can see the hypotonic solution. There is obviously more solvent on the left side than the right side. The right side has more solute. Isotonic solution is where it's the same on both sides. In a hypertonic solution, you can see on the left that there's a lot more solute than there is solvent, comparatively to the other side. Thanks for watching. I hope you enjoyed this video. Leave comments below as to what you liked, what you didn't like, and what future suggestions would be for videos. And I'll see you guys next week.